Our keynote this morning is Ana Maria Istrate. Welcome to New Zealand. Thanks for coming all the way here from San Francisco. Ana Maria is a Stanford graduate with a specialisation in AI, and I think we've heard that AI might be on people's brains at the moment, so we're really interested to see, to learn a little bit more about what you're working on at the moment. She is a staff research scientist in AI and ML at the Zuckerberg initiative. Her work focuses on developing virtual cell models and reasoning-based ML frameworks that integrate knowledge from experimental data, language, and the worlds of the models of biology. So I hope everyone's caffeinated and ready to learn a lot. I'll welcome you up onto the stage, Ana Maria, and everyone, a welcome round of applause, please.
Thank you so much for the kind introduction, and hi everyone. I'm very excited to be here, and thank you to the organizers for inviting me and for the hospitality. Really appreciate being immersed in kind of like the community here. Today I will be talking about my work at the Zuckerberg initiative, where we're really interested in this idea of virtual cells, and I'll focus specifically on reasoning models in virtual cells and how we can sort of like use AI to think through some of the biological questions that we care about. First of all, a little bit of an introduction about myself, just to kind of like know who I am. I was born and raised in Romania, where I got my love for math and science, and then I went to Stanford, where really I got deep into the math, stats, computer science, and where I got my AI and ML training. And then for the bulk of my career, really I've been at Zuckerberg Initiative, or CZI, where my work focuses on building foundation models for biology and kind of like advancing, reasoning models and this whole area of virtual cells, which I'll be introducing in a bit. To level set the current landscape, and some of the people here are probably already aware of this. Right now, we're at this place where in biological research, we have 90% of the work is done in the lab, right? And about 10% of the work is done computationally. So we're still relying heavily on experimental validation. Discovering new treatments for diseases takes decades. We can't really kind of like skip these preclinical parts. So a lot of the times we have to wait until the clinic to see that something fails. Human disease biology is very vast, right? It's very complex. So there's so many things we still don't understand. Gene function, genetic variation, mutations, all of these remain bottlenecks. There's so much of the human body and human cell that we still don't understand, and also how everything comes together. And last but not least, and this is very important, lab experiments are costly, right? They're time-consuming, they often fail, so a lot of the time you have to wait a very long time to just figure out that something does not work. So that's where we believe AI can really have, a big impact in kind of like inverting these percentages. So you go from kind of like biology from this discovery science to more of a design science. So what do we mean by that? Kind of instead of having this 90% lab validation and 10% computational, we want to be able to invert to having it like 90% computational. So you rely a lot more on simulations, and then you only validate in the lab about the things you're very, very confident about. So having this kind of like inverted ratio of 90% computational and 10% lab validation. And then kind of like what that would get us is compressing these experimental timelines from years to days. So, doing this kind of like hypothesis search in silico So we've seen all of these advancements in AI models and really, really powerful at the number of kind of like different techniques. Now, that means that we can use all of this to kind of like shorten the amount of time that we're spending in getting to either, form our hypothesis or test the hypothesis. And then there's also this idea of wanting to democratize, advanced computational cell research by integrating knowledge and tools. So there's also an aspect of just making it more widely available. So if you go, if you kind of like go from this computational angle, that also means that the resources could be more widely available to a larger population. And then the holy grail here is being able to kind of create this, if you will, digital biology simulator that's spanning multiple scales. And again, I'll touch on this in a little bit as well. But kind of like having these powerful simulators where you can test hypotheses, use what you're getting back to form new hypotheses, and kind of like shorten that overall time. So you're able to enable what-if scenarios that could be maybe impossible in physical labs. So this is kind of like the vision, and many of the reasons why I think right now there's really the time for AI in cell biology and biology in general, but for cellular biology in particular. So, there's been this paper, there's been many papers on kind of like the perspectives on how or like where AI can be best used in cellular biology, where the opportunities are. But the idea here is that you have this loop between a scientist, a model, and data. So if before it was maybe a loop between scientist and data, and now you kind of like integrate these AI models, and they're not by themselves, right? Everything feeds into each other. So basically, the AI scientist informs model development gives hypotheses back, and then that generates data. The scientist uses the data, and then we're also using data to train the model. So it's kind of like this cycle and this feedback loop. So why now, right? Why is now the moment for using AI in cellular biology? One, the scale of biological data. It's just it's at an inflection point, especially in cell biology. So far, biological data has been, slightly smaller scale. We're talking about maybe tens of thousands or like hundreds of thousands of data points. If you're thinking about the models of the scale of ChatGPT, right, like Gemini, all of these super large language models, why they're so good is because they're trained on, one of the reasons they're so good, it's because they're trained on huge amounts of data. So you could never, until now, you could not have said the same for biology. But right now we're at this point where there's a lot of community efforts and industry efforts combined to create these large-scale kind of like data atlases. And we're talking about billions of cells. So now we're kind of like it's an order of magnitude, or like a number of orders of magnitude larger than we had available so far. The second one is compute infrastructure, and AI capabilities kind of like have reached this. We call it critical maturity, and computing power is no longer the bottleneck. And what that means is that I think the biggest advancement there has been around when transformers came around, right, where we are able to parallelize easier these transformer-based models. So then that kind of like really contributed to this large advent of large language models, and then kind of same paradigms apply to other fields as well. But bearing that you do have access to this compute, basically the limitation is no longer the hardware or the capabilities of the hardware. And then, last but not least, measurement technology. So a lot of the things we care in this area of cell biology is, right, we're looking at inside cells. Again, they're very complex entities. So a lot of the bottlenecks so far have also been kind of just not being able to measure what happens inside of a cell to get this data. But there's been more and more advancements in kind of like improving the technologies across multiple kind of like axes there. So the measuring technology is getting better and better, and we're able to kind of like look inside cells more and more. So just a note on kind of like this data generation effort, because I think it's very interesting, and again, I think it's one of the key components on why this is the time to use AI for for cellular biology. We have seen these kind of like large-scale data generation efforts across the industry. So we have also launched this this program of and committed to building a 1 billion cell data set. Kind of like spanning multiple types of metadata, multiple types of conditions, perturbations, and so on. So kind of like a varied enough data set. And we're not the only ones that do that. Again, there's a lot of kind of like appetite in the industry as well, across from people interested in this area, virtual cell modeling, cell biology, to build similar data sets. So there's a number of biotech startups that also invest in this, and nonprofit partners, Tahoe, Arc Institute, if you're familiar with them. But basically, there seems to be an appetite for just building the data itself to build these models. So, like I've talked about this idea of like virtual cells, and the truth is that virtual cell modeling can mean different things to different people, because again, the idea here is just you want to be able to model cellular behavior, but depending on who you ask, they might have a different vision for that. And that's okay. There's multiple papers that kind of like talk about what the vision can be and what we envision this virtual cell, if we are to truly achieve a virtual cell, what we want this virtual cell to do. One paper that kind of like clarified things for me, and I think for others in the field, is again, this perspective paper that was published in Science that kind of like outlines quite clearly the things we would like a virtual cell to do, and kind of like what are the core capabilities, and what are the core what data needs to go in, what we would expect the system to be able to do. At its very, the very kind of like, not naive, but simplistic definition is that we want to be able to build AI-based models to predict and understand cellular behavior. So again, that can mean different things, right, for different people. There's a number of core elements that, again, the paper outlines more of those kind of like in detail, but there's a number of core elements that I think are worth mentioning. Again, you want to be able to incorporate information across scales and modalities because cells are this complicated biological entity. So, there's so many different types of information, right, that we can use, whether that's at the genomic level, transcriptomics level, like spatial transcriptomics level, imaging, all of that. We want to, molecular level, we want to be able to incorporate everything that goes on and all of this information that a cell really has access to when we're building this AI virtual cell model. So it should be able to incorporate all of this. And then you end up with what we call these universal representations. So whether we're talking at the molecular level, cellular level, you end up with kind of like this universal representations across these different scales. And then the second core element is that in this reality, when we have this AI virtual cell models, models become virtual instruments. So what do we mean by that is once we have these universal representations, we can use AI models to poke at them and to see how would the representation change under certain conditions. So that means if I have a representation of a cell of a particular cell type from a particular type of person and other characteristics, if I want to ask the model what would happen if I add a perturbagen to the cell, let's say I add a drug, or if I knock a gene down, or if I up a gene's expression, I should be able to get back this post-perturbation representation back. So it's kind of like you have this representation of a cell, you ask it your question, how would the cell react under a particular perturbation, and then you're getting back the information you want post-perturbation. So, that could look at maybe like phenotypic information, right, like post-perturbation and gene expression if you're dealing at the gene level, and so on. But the way you'd be able to do that is through an AI model. So I want to hone in a little bit on perturbation prediction, which I've mentioned it, and kind of like just say, more broadly what it means, because it is a task that it's at the core of this virtual cell modeling idea, and also a task that's at the core of the models I'll be talking about. So basically, there's two types. I mean, there's multiple types, not just two types of perturbation, but two that are the most obvious ones. Is this genetic perturbation where you take a cell and you knock a gene down, or you up its expression value, and then you're asking what happens with with the expression of all of these other genes in the cell. So this can get at disease mechanisms, right? Like you can start to understand what happens when when gene functions are kind of like turned on or off, and so on. The other type of perturbation is this idea of chemical perturbation, so drug perturbation, which is the most interesting one. Interesting in the sense that it's easier to translate, or like to get to the translational piece of it, because this is when you'd be asking, let's say I have a diseased cell, what happens if I apply this drug to it? And then you can look at the profile you're getting, and then if that matches, a healthy a healthy cell profile, then that means that the drug is beneficial, or the other way around. If nothing happens statistically, then it means the drug is not beneficial. So it's kind of like this idea of poking around the system and seeing what happens post applying this perturbation. And this is where most of my work and the work of my team at CZI has been so far, and kind of like this virtual cell modeling realm. And it's a task that will come up in the models I'll be talking about. So just to kind of like mention some of the models I've been involved in at CZI and that my team has worked on at CZI, and I'll talk about two of them today. I'll go in more detail, but one, SCGenePT, it's a model. So all of these models are models for transcriptomics data. The SCGenePT is our first kind of like CZI model in this virtual cell modeling area. It looks at combining information from experimental data and language as priors, and I'll talk about that. Transcriptformer is also, it's kind of like our first effort at really going deep on large-scale pre-training, where we trained on large-scale atlases, and it uses cross-species information. And it's really kind of like a foundation model that we tested on downstream tasks. And RBio, it's our first reasoning model that's also trained for perturbation prediction. And it's kind of like, it gets us this idea of using these virtual cell models as simulators and training on signals from them. And I'll go deeper into this as well. And yeah, if you want to learn about any of this, I'm happy to either share the papers. They're on I'm sure they're on our company website somewhere. But yeah, happy to share more resources if you want to read, go into the papers as well. So starting with SCGenePT, SCGenePT is, as I said, a perturbation prediction model that incorporates language from models of experimental data. And we showed that we can outperform other similar models by kind of like combining modalities. So the main idea here is that we have models of experimental data. So that would be some of these virtual cell models that I've mentioned, and then you have all of this information in the research literature, right? So there is so much information that's just embedded in the kind of like literature itself. And the whole idea here is, can we use both? So these large-scale foundation models that we're building, these virtual cell models, these are trained only on experimental data. So you can think about gene sequences or genomic sequences, but they're trained in the same paradigm as large language models. But except that, in large language models, we have words, right, that form sentences, and in kind of like this biological realm, we're getting around that by treating either, genes as words, and then we call them cell sentences, where a sentence becomes a cell, and then a gene kind of like becomes a word. So anyway, the idea is that we're training these large models on experimental data only. This is data, from these large scale data sets. Efforts I've mentioned, and we're training them in the same way you'd be training kind of like the GPT models and other large language models on natural language. And then there's this other piece of kind of like, again, you have information from research articles embedded in the literature itself that is in natural language, because that's how people publish papers. And then can you combine both? Does it even make sense to combine both? And the other interesting thing here is, because we care about kind of like at this level of genes, this natural language information in research articles, you can go a step further, right, to make it more organized. And there's a number of databases that basically aggregate this information. So again, maybe some of you here have worked with, or things like NCBI, kind of like a UniProt. So NCBI is really nice. They give you all of this information about genes. We particularly use gene cards. That's kind of like a summary about that gene from what's been published in the literature on that gene. So it's a nice aggregation that's also been scientifically verified. The UniProt summaries that's for gene protein coding genes. They just give you kind of like a summary of the protein that the gene encodes. And the gene ontology, which is a very, very rich resource, it gives you information about the genes at different levels. So we've used extensively molecular function, which tells you what role a gene might play inside of the cell. Cellular component, where a gene or a gene product might localize, so whether that's kind of like nucleus, nucleoplasm, whatever, and then this biological biological process. So basically, larger biological processes that the gene might be part inside of the cell. So that's to say there's multiple ways to kind of like get at what's in the literature, and one easier way it's not easier necessarily, but it makes it a little bit easier to deal with, is kind of like using these knowledge bases that are aggregation of the information that's in the research, published research. So our motivation with here has been this kind of like improving perturbation modeling, because as I said, it's at the core of the work we do. And there's two paradigms for this, and in how people usually kind of like deal with these very kind of like specialized tasks, right? So perturbation modeling is a downstream task for us. So you can think it's similar to summarization or question answering for large language models. It's just a different type of task related to what we care about. And these two approaches, kind of like here on this side, they're not specific to to biological models by any means. It's what you see across kind of like natural language models as well. You have this paradigm of you have a large foundation model that's trained on this huge amount of data, and then you fine tune it on your task of interest, right? And that's an approach that people have taken to deal with this perturbation modeling task. In our case, it's usually at one modality only. So again, you can think for large language models, you're only using at language. And actually, some people use that for biological data too, which is an interesting way. But then the most common way for us, it's people just kind of like use one modality. Let's say we're interested in transcriptomics, just kind of like use transcriptomics information, and that's it. And then the second kind of approach here is when you have a specialized task. It's you can build these bespoke models, right, for the task you're interested in. So this is where you train on a smaller amount of data, and then you embed the knowledge that you have about the task you're interested in the model architecture itself. So this is where you build very specialized models for exactly the thing you care about. And this is probably kind of like related also to the keynote yesterday, right, where we see that in that case, we had kind of like a smaller data set, and the modeling techniques were very much tailored to kind of like the task at hand. And there was a lot of kind of like knowledge from, language experts embedded into kind of like the model architecture, model evaluation, and so on. So this is where you embed your own knowledge or expertise and kind of like in creating the model architecture. And the question we were trying to answer here is like, can we kind of do that, but incorporate the knowledge through language? So we don't have to kind of like go and find the right model architecture, but basically, can we incorporate all of that knowledge that we would have, prior to in these models of experimental data using language as a means? And then the second one was, is this information useful, right? Like the premise is that it would be helpful to have all of this information from research literature. That would help our models of experimental data, but maybe it's not that helpful. Maybe the models are able to learn everything they need to learn and everything that's been published already from just kind of like learning from experimental data. So I'm going to go a little bit over the model architecture. For SCGPT, so we've taken a model architecture from kind of like one of the state-of-the-art foundation models for transcriptomics. And the way this works here, so you starting at the bottom and going up, again, in transcriptomics, we deal with sequences of genes. So again, in the same way that you'd have a sentence in natural language, and then you'd map each word to kind of like an embedding, we're doing something similar here, where we create these different representations and different embeddings per gene. So in our case, because we care about perturbation modeling, we have these what we call perturbation tokens. So if you remember, I was mentioning how one of the things we're interested in is what happens if we knock a gene down to the expression of other genes, and that's exactly what we're looking at here. So in this case, it's just a binary thing. Every gene gets a token, whether that's the gene of interest, we want to like knock it down or not, right? It's a 0 or 1. Then we incorporate these gene tokens information. So this is kind of like the yellow layer from the bottom up. This is just telling us the gene identity, right? So this is where we know this is MALAT1, this is the FOSB gene, we know kind of like what gene we're looking at. Then the blue layer is the gene expression value. We incorporate the gene expression value, so this is where we know kind of like how many counts of that genes we have. And then this is where we're adding kind of like an additional layer, this red layer, where we're adding this information from the literature, where we're adding it per gene. So this is where we're adding prior knowledge. So now you have per gene all of these different representations across the different axes. So you have the perturbation kind of like information, you have the gene expression value, the gene token, you have the language information, and you end up with kind of like all of these different representations per genes, which you can think are different types of information about that gene. And then we add all of those together, and then we pass them through a transformer encoder layer that's kind of like learning general representations about what's going on inside of that sequence. And then we pass that through a transformer decoder layer. And that's the layer where kind of like it's decoding the post-perturbation expression value. And then what we end up with at the end is this post-perturbation expression value, where for each gene, we are getting back the expression value after perturbation. So this is kind of like the idea of the model architecture we've used, which is a transformer-based architecture, and again, how we're incorporating all of this information. And to make it very concrete, right? So let's say we have the sequence of the sequence of MALAT1, FOSB. I can't really see the other one, but anyway, I think it's FOXL2. So we have this sequence of genes, and we have this FOSB gene. So we have a representation from the experimental data. This has been learned by large-scale pre-training, and then again we can query all of these different databases, right? NCBI, UniProt, the gene ontology, and then we can get additional information, about this gene. So then we can, you can read kind of like this is the gene card summary for the FOSB gene, but it tells you kind of, it tells you natural language, a little bit more information about what the gene does, what kind of like gene family it's part of, what it's doing inside of the cell, and so on. So what we do is we take this and we embed it with an LLM. We use some of the open source GPT models, and then we create an embedding, and then we add that embedding inside of this model architecture at this layer across with the other representations. So some of our key results from this project is that we found that our initial hypotheses were correct, and adding this prior information from knowledge sources through language provides additive and complementary value to just kind of like the gene representations learned from experimental data only for our task of single-cell perturbations. We've also found that using just language wasn't good enough. So if you just kind of like use these NCBI gene card summaries or UniProt, summaries or so on, the performance really lagged behind. And then that told us that having these models trained on large-scale atlases really does give us meaningful representations. So you can't really bypass them. We've also had this really interesting kind of like finding where we've, across all of the different databases we looked at, we really found different performances. So we found that different types of scientific knowledge extracted from language provide different types of priors. So very interestingly, across multiple data sets, we found that this subcellular localization from gene ontology was providing the most value for single gene perturbations. And it kind of makes sense because this is where we're telling the model, information about where a gene might localize inside of the cell. But the model, when it's trained on large-scale data, it doesn't really have that spatial information because it's just seeing kind of like a sequence of genes. So this is, I think, one where it actually makes a lot of sense that this information proved to be useful because it's something the model would naturally not see during training. And then we've also found that these protein summaries, when you're looking at 2 gene perturbations, so you can really go all out, right, with these perturbations. You can knock 1 gene, you can knock 2 genes, you can knock like 3, 4, 5. It just gets very, very complicated because then you have like a lot of interactions between how genes might kind of like, interact with each other. But then for 2 gene perturbations in particular, these protein summaries were very helpful. And the hypothesis there is that maybe, when you knock 2 genes, it's very important of like for protein coding genes, kind of like how proteins end up interacting, or like the proteins of those genes end up interacting inside of the cell, something like that. That's just us trying to justify what we've seen experimentally, right, and our best guesses. We have a lot more comparisons with models, and a lot more numbers in the paper as well. These are kind of like just the main results. So kind of like just to talk a little bit about the impact, because this was the first model at CZI that we've done in this realm of virtual cell modeling, and it was kind of maybe 2 years ago or something like that. And it was the first time that we had to kind of like really think about how to make these models open, what it means to, where should we put them, what it means to open source them. So we have this, we call it a virtual cell platform, where it's a lot. Since then, we have a lot more models, but it's this kind of like, displays an aggregation of a lot of models related to virtual cells. And then for us, it's really kind of like, it made us think, what do we need, like both internally and externally in terms of documentation, right? Like information, how to work cross-functionally across different types of like, people, how to establish practices, guidelines, and all of that. And then for us, it was also kind of like the moment where we realized that there's a lot of value in this prior knowledge and incorporating this prior knowledge with experimental data. And this was also the precursor for our larger model in this space, which is RBio, which is a reasoning model, and I'll be talking about that next. So RBio, it was started in 2025. Now it's 2026. But in 2025, we thought it's it's the year where the reasoning models kind of like really made huge advancements. Can we teach LLMs to reason and talk about cells? Can we? All of these DeepSeek papers came around, right? We've seen kind of like all of these reasoning capabilities in large language models. Again, whether it's the OpenAI models or Llama models, like the Anthropic models, all of them. So reasoning became a big thing, and then the question is like, can we use this as a means to interact with these virtual cells? And then RBio, it's a reasoning model for biology that, again, is trained for genetic perturbation prediction, which is what we care about. And the main idea here is also that we're training with using virtual experiments, and I'll explain what that means in a little bit. But before, kind of like a very brief intro to reinforcement learning. This is a very simplified schematic, but in reinforcement learning, you have this agent, right? That interacts with your environment. So the agent has access to an environment, and then the agent goes and explores the environment and takes certain actions inside the environment, and the environment, based on the actions the agent takes, gives back a number of rewards. And then kind of like this goes on for a little while or until you stop it, or you can decide the stopping conditions. But what the agent is optimizing for is maximizing cumulative reward. So that's what the agent is incentivized and trained to do. And this idea, it's at the core of, again, all of these reasoning approaches or advancements that we're seeing, which all of them have some type of reinforcement learning component in their training. So then in our case, RBio in particular, it's a model that bridges this virtual cell modeling and AI reasoning. And looking at this kind of like diagram, if in general reinforcement learning have an agent that explores the environment and gets rewards, for RBio we imagine we have this RL agent that interacts with an environment of world models of biology, or we kind of like go interchangeably, but virtual cell models of biology or just models in general with this knowledge that I was talking about prior to. And then it goes and interacts. It has access to this environment, right, of models, knowledge, scientific papers, and then it goes and it interacts with them. It gets rewards back, and then it's learning from them. So that's kind of the idea here. And then why this is a very compelling idea for us is because these virtual cell models, again, one of the key motivations for having them is that you'd be able to simulate outcomes rather than go and test them experimentally. So once you're able to simulate outcomes, you can imagine you can simulate pretty much, kind of like a large scale. So you're not really compelled by what's available experimentally. So RBio turns those simulations into training signals. And again, we're learning to reason about genetic perturbations. And the question we're asking here is, again, if I knock down gene A, what happens to the gene expression of gene B? And then it goes back to this question that's at the core of virtual cell modeling: is how does its cell change state under perturbation? I've already went over this, so I'm not going to go again. But the idea here again is, we care about this task because it's at the core of what we do, and to make it very concrete of how are we training these models. So when you're training these models, you usually have a number of you have to kind of like train them with certain types of prompt templates. So two things you're usually going to have is what we call the system prompt, which this is where you set up the persona of your agent. So we're using kind of like a prompt very related to the DeepSeek ones, because we're using kind of like a Quen model as base, so we wanted to stay similar. But this is where kind of like you're setting up your agent to take a certain personality, if you will. So in our case, we set this up by saying, this is a conversation between user and biologist. The user asks a question, and the biologist solves it. The biologist first thinks about the reasoning process in the mind, and then provides the user with the answer. The reasoning process and the answer are enclosed within the think tags and the answer tags, respectively. And then we're telling the agent, exactly how we wanted to get the answer back. So we have these like think tags, and that's where the reasoning process goes in, and then we have the answer tags, and that's where the answer goes in. And then this is very similar to how you'd prompt ChatGPT or Claude or Gemini. It's just this is also what we set up during our training runs. And then the user prompt is exactly this perturbation prediction prompt. There's a CRISPR-I knockdown of particular gene. So this ABCH is just a particular gene in a particular cell line. In this case, it's this EHBH2 is just a cell line. So it's a knockdown of gene A in this particular cell line. going to result in an increase of expression of gene B? The answer is either yes or no. So we're just asking the model to give us a yes or a no. Tell me, if I knock this gene down in this particular cell line, what will happen to the effect of this other gene? So then the question, if you go back to kind of like this idea of you have an RL agent that needs to get rewards, right? So these types of prompts are what the agent is trained on, is trained to explore. So the agent is trained to kind of like explore and answer these types of questions. The question becomes is how do you verify an answer to this? So the agent can come back and tell you, yes, no, I don't know, or yes, because whatever, yes, because X, yes, because Y. How do you verify that? And how do you emit a reward so the agent knows that it's doing well or is doing, not so well? And there's a number of different types of verification, and I know this slide is kind of dense, so I'll try to take my time with it. But basically, there's 3 types of verification we're considering. One is experimental data, so that's kind of like an easy-ish thing. It's where you have an answer for your question in a data set. You just have to, one, be aware of the data set, and 2, know how to, massage the data set or like get the data set to kind of like get your answer. The second one is predictive models. So this is where these virtual cell models come in, and then you can query models to, simulate an answer to the question of interest. And the third one is where we're using, again, prior knowledge to kind of like align the reasoning traces, and I'll talk about this in a little bit. So kind of going in order, again, our prompt is exactly this, is a knockdown of gene A likely to result in differential expression of gene B? The answer is either yes or no. So the model has to just give us a yes or a no. That's the final end goal of the model. We care about getting the reasoning of how we get there, but the main goal is to improve accuracy on this binary answer, yes or no. So then, if you have the answer to this question, right, verified experimentally in a data set, so we have some perturb-seq data sets, or some data set. We have access to some data sets where this question has been verified experimentally. So where we have verified in a lab that if we knock this gene down in this particular cell line, then yes, this gene, this other gene goes up or it goes down. Then we just have to, again, be aware that the data set exists, and then go and just look it up, right? It's a simple lookup. So then we can, if the model says yes, and the data set shows a yes as well, we can give the model a high reward, right? So we're using rewards between 0 and 1. So in our case, we get maximum reward, a reward of 1. If the model says yes, but the data shows no, then we're giving the model a 0. So that's kind of an easy one. However, that's not usually going to be the case because, again, perturbation prediction, it's kind of like you have this combinatorial space where it's the space that has been verified experimentally, it's not that large. So this is where we want to use these virtual cell models or predictive models to kind of like generate an answer. So then this is where we would go and we would query a predictive model, and then we'd get an answer to our question from a predictive model. So now it becomes, what does the model think the answer to this question is versus what does the data show? And when, presumably this model has been trained on the type of thing you're interested in. But because this is no longer something that has been verified experimentally, right? It's a prediction by a model. Now this becomes more of a probability that comes from the model itself rather than a clear 0 or a 1. So you can imagine I have a model that's trained exactly on this type of task, kind of like gene knockdown prediction, and this is exactly what we've done. And then we just query the model, and the model can give us, well, I think this is a yes with probability 0.7. So then if our agent comes back with a yes, then we just give the model a reward of 0.7 because that's what the model thinks that the reward should be. So this is kind of like RL from experimental feedback, experimental model feedback. And then the last one is it's a known kind of like thing across all of these reasoning models that a lot of the times you're going to get the correct answer with an incorrect reasoning. So maybe you've seen that, again, just by interacting with ChatGPT, Claude, whatever. I know I see it a lot. Oftentimes you'll see the correct final answer, but just kind of like a reasoning trace that makes no sense, right? It's misaligned with the final answer, and that does happen a lot. And it also happens, it's not just, again, for these large-scale models, but in the models we train, and I think everyone who's kind of like dealt with these models will be able to tell you they've encountered this. And it's not an easy problem to solve. It's when the model just kind of like diverges, for whatever reason. But it's very interesting because the final answer, again, is the correct one. So what we're trying to do here is we're trying to kind of like align the model with what we think it's accurate biological information. So again, relying on knowledge sources like the Go ontology because it's a reputable scientific resource. What we're doing is we're trying to steer the model, the reasoning part of the model, so not just the final answer, but the reasoning part by scoring the reasoning trace itself. So what we're doing is we're asking the model to tell us what gene information it's using in kind of like its reasoning trace. So if we had those system prompts, we're just adding another sentence where we're saying, please let us know what gene information you're using, and then we're scoring kind of like that gene information in the reasoning trace. We have different scoring functions against knowledge bases, and this is not necessarily to improve the final answer, although we hope it leads us there, but it's to align the reasoning itself with the final answer. And this is the last one, RL from prior knowledge. So how does this look like in practice, right? So we go through all of this, and then we ask our question, is a knockdown of gene A in this particular cell line likely to result in differential expression of gene B? The answer is either yes or no. So again, we've prompted the model to give us the reasoning trace under these think tags and the final answer. So you can see for this particular case, the model comes back and says, well, this GSDMA is a gene that encodes for a transmembrane protein. Knockdown of GSDMA would lead to a reduction in the amount of that protein. However, the effect of knocking down this gene on the expression of this other gene, C, and H, is not directly related to its function. They are not known to be part of the same gene regulatory network or interact in any way. Therefore, knocking down GSDMA in RP1 cells is unlikely to result in differential expression of this other gene. Therefore, the answer is no. So you can see it's kind of like trying to reason its way through it, right? And kind of like why, and that's the whole point here. Here we're trying to kind of like make it have this like multi-chain reasoning, or at least kind of like steer it towards that. Again, it's not straightforward to have it do that. So how do we train these? I've kind of like already went over this, but we're using these virtual cell models as verifiers. We call them soft verifiers from the virtual cell models in particular, because again, what we're getting back from them are probabilities, are not binary, binary things. So we start from a Quen 2.53 billion base model that we post-train with reinforcement learning with these models in the loop. You'll notice that this is a fairly small-ish model in the realm of all of the Quen models that are available and non-Quen models, and it actually worked really well for us. So basically, what happens is we take this base model, we post-train it with GRPO, which is a reinforcement learning technique, and the model again it's optimized to maximize the cumulative reward. And during training, what happens, we feed these prompts, and then the model will generate multiple completions. So it will say maybe, yes, final answer yes, and then it will give you some reasoning trace, and then you'll have multiple options for what the model generates. And this is where you use the three types of verification I've talked about before to generate rewards. So again, the model is trained to kind of like get the maximum possible reward, where rewards are defined by the three different mechanisms I've talked before. I'm gonna try to kind of like maybe summarize what's on this slide, but one of the key findings that we had is this kind of like reinforcement learning from experimental model feedback, and which is at the core again of why we started this type of approach, where we really show that you can use these virtual cell simulations as reward signals. So our results, if we look on kind of like, or maybe first I should say, so, we have this perturbation prediction model, which is pretty much a fancy MLP. It's not a very complicated model architecture, but it works really well for our task of perturbation prediction. So again, we're taking these types of prompts, right, and the MLP is trained to give us answers to these prompts that we care about. So again, this is the model that will give us this probability. And then we ask our question, and then we get this probability back from the model, and then this is what we're feeding as reward. And then this is also it's very interesting because this is also how uncertainty gets propagated into the model itself. And GRPO in particular, it's a technique that does really well with kind of like having these non-binary rewards. It's very interesting, yeah, if you want to look into it. So then what we've shown is that we have these RBio models trained with predictive models. We just call them RBio MLPs. And then we were able to, and COT here means chain of thought. I'll mention it in a little bit. But basically by training these models and, prompting them in different ways, we are able to surpass, the Quen baseline by large. So you can see this is just the Quen model itself, the Quen 2.53 billion. The difference is huge. And that's also kind of like something that we've heard from other people across the ecosystem, that these frontier models by themselves are really, really bad at biological tasks, specialized biological tasks, without additional fine-tuning, post-training, or anything like that. They just don't have enough biological knowledge, or they have it, but they don't know how to access it. So you need like complicated prompts. It's very hard to get there. This model Summer is a model that kind of like takes that approach. It's also a reasoning model that's based on a Quen baseline, but what they do is they take this approach of just having very, very complicated prompts, introducing a lot of prior knowledge inside the prompt itself. So you can see if you're just trying to answer this question, about the gene knockdown effect, it will include all of this information about genes, gene regulatory networks in this huge prompt. Versus we're literally just training, right, with these rewards in the loop. Again, this is quite remarkable because if you think about it, it's you're training on simulations. You're training on what models believe, to be true, and then we're showing that actually works. So that opens up the avenues because of just using simulations to train for your particular use cases. We've also seen that. Composing verifiers leads to stronger reasoning. So basically, we have all of these different ways, right, of verifying things. So we have experimental data, we have models of predictive models, we have the Go ontology. So what we've seen is that combining these verification sources leads to stronger performance. And we've also seen that the training order matters. So if we start by aligning the traces with the Go ontology, and then adding experimental data or predictive model feedback, that works better than the other way around. And that's kind of aligned with general training paradigms where, some people refer to this as curriculum learning, where you start with broader signals first. So when you do this post-training, you start with broader signal first, noisier signal, and then you kind of clean that up later on with signals of higher confidence. And that's exactly what we've seen too. And then kind of like the main takeaway here is that, in most cases, you're going to have a mix of experimental data and predictive models for the task that you're interested in, and then use experimental data for what you have, and then use predictions for what you don't have, and then training on that combination. It's really going to get the best of all the worlds. Yeah. So we also seen that we are able to improve test time, reasoning, and interpretability at inference time. So with kind of like training with GRPO and reinforcement learning, that's us trying to improve reasoning capabilities through training at training time. What we've also done is improving reasoning capabilities at inference time by using this chain of thought, where we're basically we're prompting the model to tell us what it's thinking, and this also improves interpretable reasoning traces. And we've seen that across the board, kind of like asking the model to tell us its reasoning improves performance. And how it comes down, it's maybe a little hard to read, but what happens here is, and this is a chain of thought from before as well. So we're just adding this yellow kind of like sentence to the system prompt, where now we're saying the biologist will evaluate each step of the problem through logical reasoning and evaluations from the prompt. We've tried multiple different ways of kind of like telling the model to go. Basically, this is a think step by step equivalent for us, and we've tried multiple different ways of doing this, and this is just what worked best for us. Talking a bit about the external impact of this model, because it's been really nice to see it. We've presented as an oral presentation to this AI virtual cell workshop at NeurIPS, which has been taking place in December last year. We've also seen being used by publications, including this Nature publication, where they had this pair of genes they were looking at, and then they verified their experimental findings basically with our bio findings. So that's been really nice to see how people use it. We've also seen a lot of community engagement. So we put all of our models are open source, and we have our GitHub repository, and our bio in particular. We've seen a lot of engagement there with the kind of like having the highest number of stars and things like that. And we've also seen kind of like excitement, from our founder mentioning in podcasts and things like that, and just general like media, YouTube channels, and kind of like community engagement. So maybe to kind of like wrap it up with some key learnings, we've learned that using VCMs, we know that we have VCMs to simulate outcomes. We've shown that our bio turns those simulations into reasoning signals. We've kind of like proposed these training paradigms to train from experimental model feedback, these predictive models, and prior knowledge as verification mechanisms to learn from virtual cell models. We've shown that composing verifiers improves reasoning, and we've shown that we can improve reasoning at inference time also with this chain of thought technique. These are like unpublished results, but we've shared them widely, so I'm comfortable sharing them. But we've also observed that there's strong transferable kind of like reasoning capabilities for out-of-distribution tasks. So we've taken these models trained on perturbation prediction, and we've evaluated them on something completely unrelated, which is disease prediction. And then we've seen large improvements again against the baseline on 2 data sets, one Alzheimer's, one cancer data set, in just kind of like giving the model access to a genome of sequences, the top 100 expressed genes in particular, and then asking the model, is this diseased or not for Alzheimer, or what type of cancer is this, or healthy for the cancer one. And then again, just to kind of like emphasize this point here, is like large-scale frontier models in our case, and we've done a lot more comparisons I haven't talked about, but with all of these OpenAI, open source models, the DeepSeek models, distilled version, like multiple combinations. In our case, they really haven't we haven't really found one that had decent performance on the things we cared about. So for us, the takeaway was that these do need to be, again, fine-tuned, post-trained, something like that. And then in terms of compute, while we did many experiments, most of our trained models, because we're using this fairly small Qwen 2.53 billion model, we're actually able to train on just 8 GPUs at one time, and we have access to H100s, and then that takes around like 7 to 10 days to just train one model end to end. So I've talked about this, and maybe I'll end with this, where our bio gives us an avenue to use these multiple virtual cell models, where you have this agent that kind of like goes and explores this world full of world models of biology, like prior knowledge, scientific literature, data. So the vision here is to scale this up, and that's the next step, where we want to scale this up and give it more access to more models, more data, more scientific articles. And this is really how I believe that we're going to achieve this unified virtual cell vision that we care about, because otherwise aggregating across modalities and scales, it's a very complex thing. Thank you very much. This is my contact information. I'm happy to connect with anyone here, and my email, my LinkedIn are open, and also happy to answer any questions. And connect with people. Thank you.
That was amazing. My mind is blown. I wish that I was still doing biology now with these tools available, which is exactly what you need from a keynote. So thank you. Now,
my pleasure.
Questions from the audience.
Yeah.
Yeah, go on.
Oh, so was this fine-tuned on the 3 billion parameter model?
Yeah.
Have you thought about going to a bit larger? I mean, that's really impressive.
We have, actually, and we saw diminishing returns, and then it didn't make sense to do that, but we have, yeah.
If we sort of view knowledge base and your complex of models as like a vertical defined vector space, and then your models a way for researchers to query that large pools of knowledge and really accelerate them down within that critical space. Is there a risk that disincentivizes slow research, this sort of high-risk wet work that would otherwise extend the knowledge base into blind spots of this vector space? And is that a problem if we're accelerating research in its extension fast enough that the losses there don't happen?
If I understand the question correctly, are you asking, does it make sense to kind of like use these knowledge sources even in order to accelerate science, even though there might have gaps?
Well, so say are we gambling that the large knowledge base we have already is sufficient that linear knowledge combination technology will produce a complete cell model, or are there blind spots in the knowledge base that we still need to maintain slow wet work in order to discover those diminishing niches that are actually within the vector space of our knowledge and language model combinations?
I think it's a hard question to answer, and so it's also a little bit of a chicken and an egg problem because we have scientific research articles that a lot of times is somewhat connected with the data we have experimentally, right? Because one, it's very unlikely. There's very few cases where you're going to have experimental data and able to generate truly novel findings based on something that hasn't already been in the literature already, right? So I think it's more around, well, maybe there's gaps in how the models learn from experimental data that we can fill with prior knowledge from scientific articles. Because for whatever reason, the model architectures we're using are not, able to do that. So I think it's more trying to fill the gaps in kind of like what's existent. Other than, let's say maybe a company has this like proprietary data set that's truly novel, and they could really learn like completely new science, which I think it's a very small probability that would happen, because in general, right, science moves as a whole, and I can see that happening, but it's. I think it would be very unusual. So yeah, I think it's a very interesting problem, but I think it's more around trying to fill the gaps. But the space of knowledge we're operating in, it's probably the same. We're just trying to kind of like see, where to get, what information to get from where. Yeah.
All right, one more question. Yeah?
Thank you, that was very interesting. Why do you think that adding that chain of thought boosts the performance so much?
The chain of thought? I mean, in general, it seems, it's very interesting. So across the field, like using chain of thought is just something people have realized that for whatever reason it helps. And that's something that, seems, again, to just have higher performance. In terms of why it helps, I think there's this hypothesis that, one, you're just kind of like, asking the model to dig deeper into its own learnings. But there's this whole other area of like, mechanistic interpretability and all of that. That's just kind of like, basically you're trying to get at what has the model learned and try to get the model to tell you more of what it's learned and how it's learned. So this thing step by step, it's getting at trying to get the model to tell you exactly the pieces of information it used, and then in doing that, it might kind of like improve its final answer, because I don't know, it's one, going through more steps. That's one possibility, right? Just going through more steps is kind of like a human. Once you like go through something carefully versus carelessly, so you spend 5 minutes versus 1 minute. So there's something to say just about the time you spend, because it does take longer. You're generating more tokens. So that's one aspect. But I think the more interesting one is like, why do you have a model that's not able to just give you the correct answer without going through step by step? And I think that's a more, it's a different area of research of trying to see how models learn, right? And how you're trying to query what the models learn. Yeah.
Awesome. Anna Maria will be around, so do ask the rest of those questions in lunch. And I know there's a few bubbling around. I've got my own. I was lucky enough to ask quite a few questions the other day, but I still have more. So a big round of applause to say thank you to Anna Maria for sharing all that knowledge.
Thank you. Thank you so much. Thank you so much. I appreciate it.
Thanks so much for coming. Morning tea. Enjoy. Awesome.